Welcome back. In my previous video, I discussed the basics of botrytis cinerea. I briefly touched upon biocontrol measures and included Trichoderma members as major examples of biocontrol agents. Here, I'm going to discuss them in more detail. While there are marginally better results on YouTube for Trichoderma, the actual bibliography is more complicated than botrytis cinerea. As a result, I'm working with more disjointed sources here. Don't get me wrong, Trichoderma is one of the best studied genus of fungi, but there isn't exactly a single authoritative book or review I can easily rely on. I'm relying on many more sources here. I want to say that making generalizations about an entire fungal genus is one I'm trying to be careful with. There are obviously many different species in this genus with different functions and applications. I'm going to specifically pinpoint the reviews I predominantly used in the description so you don't have to hunt for papers like I did. Several of the sources are book chapters, which makes me slightly uncomfortable. I don't like using books if I don't have to, since reviews tend to be more up to date. But some of these books are new enough, and the citations they make prove that, so I believe they're appropriate. This introduction has gone on long enough, so let's get started. Alright, cool. The Trichoderma genus consists of filamentous fungi with prominent green spores and the ability to inhabit an incredible amount of ecological niches. Or niches, honestly. This ability is the defining characteristic of Trichoderma. The Trichoderma genus belong to the Ascomyces phylum and... Oh no. Well, this is a good start. Here's the lineage that I see most often. The Uniprot website also offers more information on things like subphylums. A common motif is that the Trichoderma genus is opportunistic, but I think people may immediately assume they're pathogens because of that word. Members of the Trichoderma genus are opportunistic in the sense that they can adapt very well to their circumstances, not in the sense that they're opportunistic pathogens. Don't get me wrong, they are opportunistic human pathogens as well, but I'll discuss that more later on. Trichoderma members can inhabit anywhere there is dead plant material, and especially in the rhizosphere of plants, or really just about anywhere. Some members, like Trichoderma harzianum, are found worldwide. However, one paper suggests that the soil is not a unique habitat for Trichoderma species necessarily, so keep that in mind. One way it survives is by attacking other fungi through various mechanisms, which I will go into more detail in a bit. For now, let's talk about taxonomy. Yep, here we are again. Trichoderma has the same exact problem that botrytis and frankly a lot of fungi have. The different life cycle stages make the taxonomy confusing. To recap, a lot of fungal organisms would previously be grouped into two separate genus, corresponding to their respective asexual or anamorph and sexual or teleomorph stages. In this case, the anamorph is Trichoderma, while the teleomorph is Hypocrea. The controversy comes when supposed connections between anamorphs and teleomorphs are difficult to distinguish and species get misidentified. For decades, Trichoderma strains were identified for only one species, Trichoderma viridie. Even after this was rectified, most Trichoderma species were and are still identified mainly by morphology. There have been recent developments to make Trichoderma identification more closely based on genetics, but the results are patchy. It only gets worse. Most of the fungal diversity is represented by Hypocrea, or the sexual stage, while most of the biocontrol agents are represented by the asexual form, or Trichoderma. Some anamorphs actually have some morphological teleomorph features, while still being an anamorph technically. Trichoderma ricei, which is important for producing industrial enzymes, can actually cross with its corresponding teleomorph H. jacquariana. Similar to botrytis, Trichoderma also falls under the one fungus, one name format. For this example, Trichoderma is the one name that takes precedent over Hypocrea, even if Hypocrea isolates didn't have asexual morphologies. At this point, many Hypocrea isolates have already been renamed. Yeah, fungal taxonomy is a mess. I'm gonna stop here. At last. So the big kahuna of Trichoderma is its application as biocontrol agents. So what is biocontrol? The definition is context dependent. Entomology is going to have a different definition than microbiology. For our purposes, biocontrol is the use of microorganisms, sometimes replacing or complementing other measures including pesticides, to control plant pathogens. Biocontrol is what Trichoderma is most well known for and for which I will be spending the most amount of time describing. Where do I even begin? As I said, one of the key characteristics of Trichoderma members is the ability to attack other fungi, in addition to their ability to inhabit almost any environment. To discuss how Trichoderma attack plant pathogens is to understand how it survives in general. When I say attack other fungi, I mean mycoparasitism. Mycoparasitism differs in mechanism depending on species. Mycoparasitism is a fancy word that describes attacking fungi, basically. There's also hyperparasitism, which as far as I can tell is just an angrier version of mycoparasitism. Mycoparasitism apparently starts when Trichoderma senses plant pathogens through chemical signals, likely through signals like oligochitin. Trichoderma interacts with the target, most likely through lectin-like proteins or something similar. You don't need to know that, and frankly, the lack of good sources for the initial phase of mycoparasitism is shocking, though I guess it shouldn't be for one of the most studied groups of fungi. So, Trichoderma finds a target and makes contact. Next, Trichoderma coils its hyphae, which are the long filamentous tubes that make up fungi, around the target and form structures that have been variously called papillae or aprosoria-like. These terms are used so frequently, but they're so poorly defined. They basically mean any point at which the hyphae penetrate the host. Next, Trichoderma releases cell wall degrading enzymes, or CWDs. Not going to say it. These CWDEs, as their name probably indicates, degrades the cell wall of the target and kills it. CWDEs include enzymes such as chitinase and glucanase. These are important since fungi, which are the most common targets of Trichoderma, include chitin and glucan linkages in their cell wall, so it makes sense that those linkages would be targeted. The targeting of a fungi through attachment and degradation is one of the several methods by which Trichoderma species are useful for biocontrol purposes. Owing to the fact that Trichoderma members are very good at taking up nutrients, Trichoderma members can compete very effectively with plant pathogens in the soil. Trichoderma species compete for nutrients, especially carbon, nitrogen, and iron. Carbon was given a special mention in one of the reviews I'm reading, especially against Fusarium oxysporum. Trichoderma also releases siderophores, converting insoluble ferric iron into soluble ferrous iron, and depleting available iron away from plant pathogens. Going any further means going into detail about metabolites, which I'm going to do right now. Antibiosis is the association between two microorganisms that is detrimental to at least one of them and that is caused by the release of metabolites or cell components. The sheer amount of metabolites from Trichoderma is kind of amazing and difficult to group together. Trichoderma species release a large variety of secondary metabolites. One example is harzionic acid, which, in addition to being a siderophore, is directly antifungal. There's also trichorzanans and trichorzans and tricholongans and longibracans and... There are so many. 6-pentyl-2-pyrone smells like coconut and is often shortened to 6-PP, or 6-PAP. I'm going to stop. One of the reviews I've been reading and citing makes it easy by specifying 3 types of the most common metabolites: peptidyls, polyketides, and terpenes. I'm going to briefly go over their structures and significance. Saldahano et al. In their chapter Enhanced Resistance of Plants to Disease Using Trichoderma Species in the book Biology and Biotechnology of Trichoderma, I have to do this, I'm citing it directly, describe peptidyls as composed of around 5 to 20 amino acids. They are also able to form voltage-gated ion channels and are synthesized by non-ribosomal peptide synthetases, or NRIPs. I'm calling it that. Polyketides are a big-ass group of secondary metabolites produced by polyketide synthetases. The IUPAC Gold Book defines the polyketide structure as containing alternating carbonyl and methylene groups. Terpenes are a class of organic compounds composed of isoprene or C5H8 units. I would go into more detail about their function, but quite honestly, it's just very individualized and would take up too much time to discuss. And these are the 3 compounds that are mentioned by this review. All 3 compound classes are secreted by Trichoderma species and are antifungal to plant pathogens. There are, of course, dozens or even hundreds we could talk about, but we don't have that kind of time. Not all Trichoderma members produce the same metabolites. Trichoderma atraviridae produces 6 pp, but T. virans and T. ricei don't. That's it. Next. Trichoderma attempts to colonize plant roots as part of its initial induction of plant resistance. Cysteine-rich hydrophobins such as TASHID1 and KID74 allows Trichoderma species to colonize plant roots using Aprosoria-like structures, here we are again, though they are usually limited to the first few layers. Expansin-like proteins like swolenin and CWDEs like endopolygalacturonase help to colonize roots. Trichoderma may induce plant pathogen resistance in plants through changes in calcium and reactive oxygen species like hydrogen peroxide. Trichoderma can also induce genes involved in the salicylic acid, jasmonic acid, and ethylene signaling pathways. These pathways are often involved in the immune response of plants. Different Trichoderma species may also induce different pathways involved in either systemic acquired resistance, SAR, not that one, which is typically associated with pathogens and induces SA signaling, or induced systemic resistance, ISR, or ISR, which is associated with JA or ethylene signaling. Trichoderma species can assist with plants more directly by promoting plant growth in various ways. Biomass, yield, nutrient availability and uptake, photosynthesis, and abiotic stress resistance, to name a few. In terms of biotechnology, our good friend Trichoderma ricei is a primary example. Specifically, cellulases are produced at an industrial level from Trichoderma ricei. Trichoderma ricei is also used as a benchmark to test new industrial cellulase strains. The oil crisis of the 1970s pushed governments to find alternatives like biofuel, and Trichoderma ricei was a primary example of that. Trichoderma ricei obviously isn't the only species to produce cellulases or use it in an industrial capacity, but it is the most used. Trichoderma has also been used in bioremediation, biofuels, winemaking, and brewing, just to name a few examples. I want to get this out of the way, since I don't care about medicine. I mean, imagine actually doing it as a career. Like I said a billion YouTube years ago, Trichoderma is opportunistic. One paper I was looking at by Sandoval Dennis et al. Explained that while Trichoderma infections aren't common, they are emergent, meaning they are increasing in prevalence. Infections tend to be caused by Trichoderma longibrachiatum, but other species that are relevant to biotechnology and biocontrol include Trichoderma ricei and Trichoderma harzianum as also occasional pathogens. Sandoval Dennis et al. Gave 3 risk factors for Trichoderma infection: peritoneal dialysis, organ transplantation, and hematologic disorders. While fatalities are obviously rare, it does happen. Other fungal species, especially Aspergillus and Candida, are more prevalent causes of fungal infections, but it shouldn't be ignored that Trichoderma can also be a human pathogen. I feel like bad taxonomy is going to be a running theme. Trichoderma is a very important biocontrol agent and a producer of industrial enzymes. It has many qualities that help plants in many ways, including plant growth and disease resistance. It shouldn't go unnoticed that it's also a cause of human illness. Bye.